Today is session 5, and we're going to be looking at different types of studies that can be conducted with depth electrodes and some of the utility of being able to now do single unit recordings with these depth electrodes. I'm basically going to talk about 3 different types of studies that are being currently conducted with these electrodes. And obviously, this is not the full breadth of what can be done, but just to give you an idea of the type of research being done. For instance, there's functional anatomy research being done using our depth electrodes. Basic brain function, people are studying different aspects of brain function, and also looking at disease state. Obviously, our patients have epilepsy. So is there anything we can garner from these intracranial electrodes from a research standpoint to understand epilepsy itself? So from a functional anatomy standpoint, we've been creating these beautiful colored charts, which essentially look at connectivity within the brain. So this green area, if you follow the green, is connected to other green traces. And the reds are connected to where there's red located in the brain. However, these are computer-generated models based on different characteristics of the MRI. And although they look cool, they have the ability to potentially help us understand encodes information that can help us with diagnosis and potentially guide surgical interventions. But there's always been a question, how accurate are they? But how do you test this? SEEG could actually help in this. In the case of SEEG, we're implanting electrodes throughout these brain networks. And what we can do in this case is to actually stimulate and record within this network to see if the green is really connected to the green or if it's connected to some other region. We apply a stimulation pulse. We look at the evoked activity. We stimulate it here. And if you follow the green fibers, we're recording the evoked activity following the green fiber. There's also the possibility, if these maps are not very accurate, that when you stimulate, you actually activate something that wasn't supposed to be part of that network. And thus, using these stimulation and recording experiments, we can actually validate our computational models of the anatomical connectivity charts. They can lead to modification of the charts that better represent how the brain is connected, or we may find how well they do work, and either answer would be of great interest. In terms of brain function, most of my research when I was still in the clinical arena was looking at decision-making processes. However, there's a variety of other things that people look at: speech processing, novelty, how's the brain encode something new that it's never seen before versus some object or something that it knows well. It can help you understand learning. So what brain areas are involved in learning, i.e., this object means a certain thing, and how does the brain encode the meaning of objects or maybe what they're associated with? We can also look at motor function. I've done some of these studies, but there's a number of people looking at this. So how is the brain encoding movements? How is the brain encoding movements when some unexpected perturbation occurs? There's a lot of questions that can be asked. And the SEEG patients provide such a wealth of knowledge because we're implanting across vast networks in the brain. And traditionally, we look at the local fields, but now as we advance these micro contacts to where we're seeing single units, we can start looking at these questions in SEEG patients at the single unit level and at the network level at the same time. I'm going to describe a decision-making task that I worked on. And this particular example's from a patient that was undergoing implantation of brain leads for psychiatric disorder. And during the implantation of those leads, we record it through an inner region known as the nucleus accumbens. And most people know this area. It's involved in motivation, its association with drug addiction and other disorders. And here we were looking at how is it involved in normal decision-making processes. So to do this, we had the patients play a simple game very similar to the Warcast that everybody's played when they were younger. Essentially, they would be shown a screen with a dot saying the trial started. The next screen would show them a card face up. That's their card, with a card face down, which is the computer's. And then the next screen would show a bet, and they had to choose a bet of either $20 or $5. So if they thought that their card was stronger than the computer's, they would bet 20. If they thought it was weaker than the computer's card, they would bet 5. After a delay in their choice, they would see what they had relative to what the computer had. In this case, their 6 was greater than the 4 that the computer had, and it showed them, hey, you won $20 because you bet $20. If in the scenario that they lost, it would say you lost $20 and there'd be a cross through it. Again, these were done single unit recordings. So this is just an example of how the electrodes were passed into the brain. A single unit recording for DBS, we use micro electrodes with one recording tip. Often we only use one electrode at a time. Sometimes we can use more. But unlike the MME can have up to 12 micro recordings at the same time, as opposed to a single. Again, we would record the brain activity and be able to sort out single neurons based on their morphology and amplitude. Obviously, if you don't get good recordings, you can't do this. However, with good quality recordings, then you can take the activities and line them to different aspects of the task. So going back to the task, this is what the patient would see on the screen. They would see a 6 card, the computer's card is down. They would make their bet. In this case, they beat the computer, and if the patient had chosen the $5, they would see this. Otherwise, they would see something like this. Now, to be able to analyze this task, because we're in the OR and we have limited time, we can only conduct tasks that are of a certain length of time. In the EMU, you're able to do longer tasks because the patient's awake. You're not interceding in a surgical arena. So you can run tasks that are more complicated. So we can actually analyze the data and not extend the length of the surgical time. We actually used only even number cards and no face cards. So the choices were a 2 card, a 4, a 6, an 8, and a 10. So the 6 card, they had a 50% probability of beating the computer. With the 2 card, unless there was a match, the computer matched with a 2, they were going to lose. On the 10 card, they were going to win unless there was a match. And then obviously the 8 and the 4 fell in between. So these are fixed probabilities, where the 6 card is a 50% probability of win, the 10 is 100% win, and the 2 is 100% loss unless there are matches. And if we now look at this image, we can see the trajectory. The nucleus accumbens is located down where this red dot. So we would actually insert the electrodes until we were in the accumbens region. Then we would actually start the behavioral task and record the single unit activities. If we look at a betting, how the patients bet over course of playing this game, this is what we would kind of expect. So when the expectations of winning are high, 100% in the case of the 10, and the expectation of losing are 100% in the case of the 2 card, your choice in betting is very simple. You know, with the 2, you would bet $5. With the 10 card pick, they would very quickly bet that they were going to win, and they would bet at $20. However, in the middle, they have to make a decision. Are they going to win or are they going to lose? And you can have different strategies. And that thought process actually slows down your reaction time for picking whatever your choice would be. And that is the 5 or the 20. If we look to see how they bet, their betting strategy actually follows roughly the probability of winning. So when they saw a 10 card, it was almost 100% probability they were going to bet a 20. When they saw a 2 card, they rarely, maybe by accident, bet a 20. But you can see on the 8 card, they bet high almost 100% of the time. On the 4 card, they didn't bet high most of the time. And then on the 6, we're near this 50% probability. That is, around 50% of the time they would bet high, 50% of the time they would bet low. But what was the brain doing during this? And this is what's really interesting. So what we did was we looked at the 6 card, where the patient's betting high or low about 50% of the time. And we looked how the brain's activity fired relative to different aspects of the task. And right here, the 0, the brain activities are lined up on when they were shown the 20 and the $5 bill. In this case, the brain's activity were lined up when they actually physically pushed the button. And we took all the firing rates when they bet high versus all the firing rates when they bet low, and the high bets were the blue line. So the firing rates were high starting before they even were given an option whether they were going to bet high, and continued to be higher than the firing rates when they bet low. So in essence, the patient's decision to bet higher and low is actually encoded by the neurons before they physically make their choice. Out here, where they physically made the choice of high or low, the activities are back to about roughly the same. So what this shows is that the brain actually encodes expectation of winning before a movement occurs. So the decision process on the movement is actually out here. Very interesting. So now we can actually tell before a patient chooses whether they're going to choose high or low just based on the firing rate of the neurons. Another interesting finding can be found on this screen. This part of the brain is thought to be involved in learning and encoding expectations. So we have 4 potential expectations. A patient could have an expectation that they're going to win. They could also have an expectation that they're going to lose. And the expectation can either be matched or not matched by the behavioral task. So we have a situation where in the green on the top screen, and time 0 is here when they were told whether they won or lost. So in the green here, this is when we plotted the firing rates of the neurons when they had an unexpected win versus the firing rate of the neurons when they had an unexpected loss. And what we can see in this scenario is there's a statistical increase in the neurons firing rate when they had an unexpected win. Likewise, when they had an unexpected loss, it actually statistically dropped. So what this is telling us that this part of the brain is involved in updating future expectations or dampening expectations of losses. So when they have an unexpected win, it will update their understanding of the probabilities related to unexpected wins and losses. It will dampen their expectation. If we look at the firing rates when their expectations were met, there was no change in firing rate. So when the expectation of loss and you lost occurred, the firing rates didn't really change. When you had an expectation of win and you won, they didn't change. So really, it also shows this part of the brain is involved in encoding future expectations. So what does SEEG provide? We did these studies doing single units in the OR. And as you can imagine, one recording at a time, your yield in neurons is quite low. However, SEEG patients, they're in the EMU. You could run this task a number of times. You could change how the task operates and see how the brain changes with that. But essentially, the beauty of SEEG is we're implanting networks, and we can see how whole networks are functioning now during this task. And in fact, we actually ran the same behavioral task in our SEEG patients. And this is just one of the publications we had. It's called Lucky Rhythms in the Orbital Frontal Cortex. Because what we found is that this part of the brain, you can actually see how the brain is biased during decision making. And I'll show you the results of that. So again, this is our behavioral task on the top. This is how the patients selected bets. So on the 8 and 10 card, as you expected, they would bet very high. On the 6 card, they're kind of betting in the middle. And then the 2 and the 4 card, they didn't bet high, hardly any of the trials. The reaction time, again, followed this curve where the decisions are being made in the middle. So the reaction times are slower than they are on the periphery of the 2 and the 10 card. And what we essentially found, and it stood out quite dramatically, is the part of the brain known as the orbital frontal cortex, which we know is involved in what's called valence, assigning a value to different objects. So this is a high value object, and this is a lower value, whether it's a positive or negative reward. We know it's involved in that process. But when we ran the behavioral test, we found a very interesting thing, and these spectrograms show it. So in these time spectrograms, we align the activity to some behavioral event, and on this axis is a frequency. So what we can see from these types of spectrograms is how different frequencies react during a behavioral task. And in this task, we actually align the start of the trial here. So time 0 is here in this case, and we're looking what happens before the trial even starts. And when we line up the frequency activities in this fashion relative to when they bet on a high on a 6 card relative to when they bet low on the 6 card. So when they bet low on the 6 card, the frequency spectrum and how it reacted in time of the OC was quite flat when they bet low. However, when they were betting high, we could see this very prominent red area. And we have a statistic to show that it's statistically modulated here relative to these two. And then if we just sum up the tight frequency range, the high bets are in the blue and the low bets are in the red. And what this shows is statistically, they're different than each other, the power. What is absolutely amazing is this occurs during the process of unexpected wins. So let's say you're on a lucky streak of betting. This activity will grow higher and higher and higher. And when you're shown a 50% probability card, you're more likely to bet high. If you're on an unlucky streak, the patients were more likely to bet low on a 6 card. So what this showed is when the patients are on a lucky streak, you're more likely to bet high, and that the OFC encodes what was called a gamma band activity prior to even the start of the trial. So I guess in a scenario, let's say you're in Las Vegas and you're playing cards and you're betting high and you're betting and you're winning and you're very excited. We would expect the OFC to actually encode that. And that excitement then could bias your decisions in the future. So you're on a lucky streak. So you keep betting high, think that you're going to win, even though you may be in a situation where you're likely to lose. So it's rather amazing, and that this happens between trials. What else is interesting is the power of this gamma also fluctuates with your heart rate. So while you're on a lucky streak, your heart rate variability is increasing, and so is gamma. And they're definitely temporally locked. Disease states. What can we look at with disease state? What is the genesis of seizures? Where is it located? How do single activities of neurons relate to LFPs? How do single neurons versus local fields react to stimulation? These are all very important questions in epilepsy and things that you can actually study with single unit activities off of the micro context, as well as the macro context. This is just an example of a study that I was involved with when I was in Boston. And in this study, what we did was we recorded from the brain, from electrodes that were pushed into the cortical layers of the brain. Then simply what we did was we templated the neurons, and then we aligned their activities to this common local field activity known as intraictal spikes. These are spiking activities that we can see on the macro level. Now that we have the single unit electrodes in, what are the neurons doing during this? Are they all firing at the same time? Are they all inhibiting at the same time? What is actually the underlying single unit response to these very common patterns that we can see in local fields in patients with epilepsy? Well, when we do the alignment, this is just an example of one neuron, and the red here is showing the average intraictal spike. And then on this part right here, every time there was an intraictal spike, and in this case, there were around 60 of them, we looked at the activity of the single neuron firing relative to each one of those intraictal events. And we can create this type of figure. This is a raster. And then if we average the activities over time, we can see right before the intraictal spike, there's a massive increase in this neuron's firing rate. In fact, it goes from, sub 10 spikes per second to nearly 150 spikes per second around the time that the intraictal spike occurs. Each hash here shows how the neuron fired relative to the spike. You can see there's some basal activity, then a rapid firing right around the intraictal spike. Then the neuron slows down and has some activity. Again, the next spike, we have a rapid increase in activity. Over here just shows how we templated the neurons and the basal activity of how the neurons fire. This is a great utility. We showed, hey, in this case, this particular neuron increased its activity right before the intraictal event. However, when we looked across many neurons, we found essentially 5 dominant patterns. And although I have 6 up here, the 5 were predominantly the main features that we saw. So what we saw is some neurons actually would fire and then have a slight drop off after the spiking event. Some neurons fired rapidly, as we said, during the intraictal events. This neuron here had an increase at the event and then an inhibition. This neuron actually had an inhibitory event. This type of neuron actually had a delayed excitation. So after the event, it fired. And here's an example of an excitation and an inhibitory event. So what this reflects is neurons are doing all different types of things during these spiking activities. Before we did single unit relative to this, we didn't really know. And now this paper actually provided some very initial evidence of what single neurons are doing during these intraictal events, which we commonly see in local field recordings. Now we can dive down at the single unit level. Are these neurons inhibitory neurons? Are they output neurons of the cortex? Are they in various layers, the thalamic connectivity layer of the cortex? Are they in cortical regions of the cortex? Now we can start diving into where these neurons are and how they're involved in this kind of very well-known activity at the local field level. So that's all I have looking at activity. Obviously, you can go out to the literature and read all kinds of exciting papers and different types of studies that other people have done. But I just wanted to give you a flavor of the range of type of research that people are doing.